Hello, everyone. Welcome to today's webinar, Automated Drug Adverse Events Detection from Unstructured Text. I'm here with Julio Bonis, data scientist at John Snow Labs. And I'm going to turn it over to Julio in a second here, but just wanted to go over a few housekeeping items. So everyone is currently on listen-only mode. However, we do strongly encourage your questions. So please type those questions into the GoToWebinar control panel throughout today's webinar, and we will be answering all questions at the end. If you have any technical difficulties, please shoot me an email and I'll try to help you out. My email is on the screen. And today's presentation is being recorded, so you will receive that in your inbox in the next couple of days. And at this time, I'll turn it over to you, Julio.
Okay, thank you, Julia. Thank you, everyone, and welcome to this webinar about identification of adverse drug reactions in structured text. I'm Julio Bonis, and I will inform you and talk to you about how this technology works with the state-of-the-art technologies. So 150 years ago, there was a lady just 15 years old that was being required apparently safe surgical intervention in one of his toenails. And on that time, doctors used a new drug class that was anesthetic, like ether or chloroform. Unfortunately, in this case, this lady died during the apparently safe procedure, and that was the start date for what we call now pharmacovigilance. And the first time doctors were aware of the importance of drug safety. Since that date until today, there has been many different milestones in the area of pharmacovigilance. We can talk about the Federal Food, Drug and Cosmetic Act in the 30s, about the Yellow Card in the UK in the 60s, the European Medicines Agency in the 19th, and so until today, where we have very complex and probably very good systems to monitor the safety of drugs. Any talk about drug safety cannot avoid to talk also about the thalidomide tragedy in the 50s and 60s, where one drug, this thalidomide, that was being marketed for pregnant women as an antiemetic, had been tested in animals in clinical trials, but had never been tested in a broad population. So when this drug started to be used in pregnant women, some very severe abnormalities started to arise. And this is the original letter of a doctor, Dr. McBride from Australia, that was one of the first doctors that had concerns about the safety of this drug. And at that time, there was not monitoring systems after a drug was approved, so everything worked like this with letters from doctors and so. After this thalidomide event, new systems were put into the system, so the drug safety was monitored not only previous to the approval of the drug, but also in post-marketing surveillance. And right now we have a lot of very complex systems that are controlling or making our drugs more safe. And the new trends in this area, probably we're going to talk about a broader concept of adverse drug event or adverse drug reaction that now is considered as response to a medicinal product which is noxious or unintended. Something that is new in the recent years is also the greater involvement of patients and citizens in these pharmacovigilance activities. And now the regulatory agencies want patients to report when they have a suspicious of of an adverse event. And also, of course, now we have what we call big data. We have tons of data available. So we need somehow to find ways of automate the detection and treatment of these safety issue signals using secondary sources of data as electronic healthcare records and social media. If you are here in this webinar, it's because you know that adverse drug reaction is a very important issue, but here we have just telling you some numbers. We know that it's the single largest contributor to hospital-related complications, affects more than 2 million hospital stays annually, prolonging the hospital length in 2 to 5 days. It contributes to an additional $30 billion care costs in the US, is the cause of 5% of all hospital admissions, and just in Europe yearly, it is responsible for an estimated about 200,000 deaths just in Europe. So it's a very important issue. Right now, we have different pharmacovigilance strategies. We have this monitoring during clinical trials. In clinical trials, every patient is very well monitored, so we are able to detect early problems with safety, but keep into account that once the drug is approved, in the next 16 years, about 1 of 4 of the drugs will be removed from the market because of the safety issues. So just monitoring clinical trials is not enough. It's clearly not enough. And then we have these post-marketing surveillance systems that can be classified in 2 kinds of systems. The first ones are spontaneous support systems with the well-known databases, fires, vigilance. But those systems have the problem that most of the adverse drug reactions, even the serious ones, are usually detected but never reported. Up to 85% of these events are not reported. So we have a problem of underreporting. And then in recent years, we have this, what we call sentinel programs that will scout over electronic healthcare records and social media, and will try to identify these adverse events. But the problem with these systems is that we know that about 70% of the adverse drug events are not registered in a structured way, but are registered in clinical notes and or other unstructured data that we need to process. Also, we need to always keep in mind that in the realm of adverse drug events, we have those on the top that are noticed and reported, that are probably a very small portion of the events. Then we have those that are noticed by the patient or doctors that are unreported. And then we have a huge amount of them that are simply unnoticed. And those are these events that are mostly long-term causal mechanisms. You take the drug and then you develop the problem, the health problem, years later, so you don't link the drug with the problem. Those that have a non-pharmacological effect modifier, maybe you take the drug and only if you take frequent sun baths, you will have a problem. If not, you will not have, so you don't link it. Or where in that case of events that are linked to pharmacogenetic mechanism, or in events that are related with polymedicated or medicated patients or patients with many morbidities, this is difficult sometimes to identify the problem and link the problem to the drug. Regarding these recent efforts to identify adverse drug events in social media, I just put this slide because I think it's a very interesting one about the kind of sources of information, sources of data that are currently being used or explored to try to find these events. You can see here that Twitter is this general social media platform. It's very commonly used in this kind of studies. And then you have many specific domain platforms that are used for that. This is a very nice example. Another nice example of this kind of approaches is this one, where the researchers identified an interaction between paroxetine and pravastatin that was linked to increasing the risk of hyperglycemia, and this interaction was reported in 2011. So they tried to check what patterns of web search happened 1 year before, before that interaction was even reported. And they found that patients that were searching for just paroxetine or just pravastatin, so they were not, theoretically, they were not combining both drugs, the number of the frequency of search for hyperglycemia was about 50%, 5%. But in those patients that had searched for paroxetine and pravastatin during the previous year, they found that the frequency was double. The frequency of hyperglycemia searches was double. So it suggests us that there is some kind of signal in this social media or patterns of use of the web from patients that can help us to identify some safety issues before even we are able to report that issues in pharmacovigilance traditional studies. Also, a nice example is this one from these researchers from Arizona, where they compare the frequency of adverse events reported in the official documents, and they compare that against the frequency of these adverse events appearing in some open forums in internet. And they found that for every drug, usually you will find the same adverse effects and in a similar proportion. So it looks like content generated by patients in social media could be a good approximation of the real adverse effects risks for a given drug. But how can we deal with this unstructured information? How can we extract and classify this information? There are many approaches, and those are mostly named under the label of natural language processing. And here today I will show you one specific library that I collaborate with as a developer, that is a Spark NLP, that is based in Spark technologies, and in fact is the top one in terms of frequency of use in the industry. Not in academic research, people tend to use other libraries, but when we talk about scaling and putting that into production, Spark NLP is probably the first option, as this survey tells us. So Spark NLP, you can use Spark NLP with different languages. We have APIs for different languages. You can use Python, you can use R language, and you can use Scala, that is the language used in Spark in general. And here today I will show you some examples using Python code, but you can use any of these three languages if you want. So basically, to use Spark NLP, you just need to import Spark NLP, and you will find two versions. You will find the open source version that you can go after this talk. You can go and download and install in your system and start to work with. And then you have the licensed version that includes some specific pre-training models for healthcare that we are importing here with the Spark NLP GSL. Then we usually need to import any other functions from PySpark or from Pandas to be able to show the outputs in a nice table and formatting. So basically, this library is very easy to use. You just need to start a Spark session with this command, Spark NLP GSL start. And in the case of the license version, you will need to provide with your license number here in wildcards, but in the open source, you don't need to provide anything. You will start just the Spark session. Well, for those that are unfamiliar with Spark works with the concept of pipelines, where for machine learning, where you will build different pipelines that will process data in different steps. So we will talk about pipelines a lot in this talk. On the left, you can see what will be a pipeline, a Spark NLP pipeline. Now, probably you will think that this is a very complex thing, but don't worry, because in the next minutes we will analyze step by step, and then at the end of this talk you will be able to understand this code and to build your hopefully to build your own pipelines for your specific use cases. So about the anatomy of a pipeline, a Spark NLP pipeline, we can split this in three main components. The first one is the blue one, where we will basically define the components of the library that we want to use in this specific pipeline. And we are building here a pipeline that will classify any unstructured text in two types: adverse drug reaction related text or non-related text. So this will work like a filter to classify documents, tweets, or whatever text that you feed into the pipeline. So in the blue portion, you will define the components. Then in the green portion, you will ensemble the different components in the pipeline. And then in the yellow portion, you will basically train or feed this pipeline, and you will generate what we call a model that then you can use to process new data. Regarding or focusing in the components, in the part where we are defining the components for this classifier pipeline, we will use 5 components. The red one is the document assembler that will take the text and will generate what we call a document and a SparkNP document. Then we will apply the green one, that is the tokenizer, that will take this text and will split it in what we call token in NLP. That is something similar to words. Then the next one in purple is BERT embeddings that we will explain later. Then we will have the sentence embeddings in blue, and then finally we have the component that is the classifier itself that will tell us if this text is related with the ADR or is not related with the ADR. For every component, you can see here in green and in yellow, every component as a pipeline will have an input. In the first one, in document assembler, the input is text and will produce an output. And the output in document assembler in red is the document. And the next component, the tokenizer, we have the document as an input, the token as an output. Then the next one, the BERT embeddings, will have 2 inputs, the document and token, and will produce an output embeddings, and so this is how it works. It's always the same structure. On the bottom, you can see a diagram that is representing this input and outputs, and these different components or annotators, what we call annotators, and how they work and how they take the text from the beginning and are passing the different outputs to the next components, and finally are producing the green one, the classifier, is producing the classification to us. Once you have defined these components, the next step is to assemble all these components in the pipeline. You can see that in this piece of code. And then finally, here we are generating an empty data frame in Spark. This is the way you define an empty table or data frame. And then we will pass this empty table into this fit method or fit function that will generate from the pipeline, will generate the model. That is what we will use to classify texts. So once we have assembled the pipeline, trained the pipeline with an empty data frame, and we have the model, we can basically take, let's say, 4 different sentences. The first one is, I feel a bit drowsy and have a little broader vision after taking an insulin. That is clearly an adverse drug event sentence. Then the second one is, I just took an Advil and have no gastric problem so far. This is talking about the drug, is talking about a health problem, but as the patient said, I have no gastric problem. This is not an adverse drug event. So this is without adverse drug event. Then we have another one that is maybe could be an adverse drug event, is I have been using aspirin daily since 96, and since then I have been feeling sick. So maybe could be an adverse drug event or not. And the final one that is unrelated completely with any drug or with any health problem. So it's coming from a report from radiology. The brain TC did not show any masses or anomalies. So we take these 4 sentences, we put these 4 sentences in a Spark table or Spark data frame with the following command. So now we have what we call the real data, the data that we want to process, and we can show it in Spark, and then you will get this output that is basically a table with a column named text and the 4 different sentences. This is the input for our pipeline. Now we can take this real data frame, and they will pass this data to our ADR model, classifier model, with the function transform, and we will store that output in a data frame that is named results. So if we show the results, that is the output of the pipeline in Spark, we will see this table. And if you see this table, the names of the different columns are mapped to the different outputs of the different components of the pipeline. You see that we have the text, that is the input, then we have the document, then the token, the embeddings, the sentence embeddings, and the class. We will now analyze each of these columns to understand better how this pipeline works, so you can understand the internal functionalities of this pipeline. If we want just to check the text and the final classification, then we can use, for example, this typical filter in Spark that is using select, and you are selecting the text column and you are selecting the class column, and inside the class column you are selecting the result. That is one part of this output. So you will show that the model has classified the first sentence as true, as related with IDR, so it's doing well. The second as false, the third as false, and the final as false. So the model is mostly working pretty well. The first component is the document assembler, and it will produce a document that is something that has no interest. It's just taking the text and put it into a format that the Spark NLP will know how to work with. And then we have the tokenizer in green, and this is interesting to check, especially for those that are not familiar with NLP. The tokenizer will basically split any text in tokens, and tokens are similar to words. And we can see here what is the content of the token column for the first sentence. And you can see that we have for the first sentence, we have like several different tokens or words. The first one is I, and the second is feel, then bit, drowsy, and have a little blurred vision after taking an insulin. This is how it is split in different tokens. And you can see here that something interesting to notice is that we have for every token, we have the begin and the end. And it represents the position of the token inside the sentence in terms of the relative character. So the center to be a token inside that sentence. What about the next component? The next component is BERT embeddings. For those familiar to NLP, this could sound like very basic stuff, but I don't know what is your background, so I will explain. Embeddings is a way of representing natural language components, like a word, representing its meaning, its semantic meaning, in a way that computers can deal with. In this case, we want to represent a given word, the meaning of this word, represented by a list of numbers, a list of float numbers. In this case, we are using BERT, this pre-trained model. You can see here that we are loading, in the first command, we are loading this BERT embeddings, pre-trained, and we are loading a pre-trained model that is available in the open source version of the SparkNP that is called BioBERT PubMed base cases. And this model has been trained with hundreds of thousands of PubMed abstracts that contains a specific biomedical domain language. By analyzing this content, the model has been able to identify the meaning of each word on that corpus of texts, and will represent any word with a list of 768 float numbers. That is what we see here. So for the word I, the first word in the first sentence, the token I will be represented by this long list of numbers. We can have another example, for instance, that this word insulin in the first sentence that will be represented by a different list of numbers. Those words with similar meaning will have similar numbers. This is the idea behind embeddings. Something to remark about BERT embeddings is the following. In the first embedding models, the first ones like 8 years ago, something like that, like word2vect or growth, those were models that were trained in a similar way, but that for every word they will provide just with one representation. So in case you have one word like bank, it will provide with just one representation in numbers. The problem with bank is that bank could mean different things depending on the context. So you can sit in a bank, or you can put your money in a bank. So depending on the context, this single word will mean different things. In the first versions of embeddings, they were not able to deal with this polysemy of words. But in BERT, specifically by how it is trained, is able to assign a different meaning depending on the context where the token appears in the sentence. And we can see this here in our example, because if we take the I token in the first sentence, it is represented by this list of numbers starting from 0.10, then minus 0.4024, and so. But if we take the I in the second sentence, that is in I just to cannot build, the representation is similar, is close, the numbers are very similar, but it's not exactly the same. So what it is saying us is that any word will capture also the context of the word to represent the meaning. We are representing not only the meaning of the word, but the context of the word. So once we have move each token, each word into a representation in numbers that the computer is able to deal with, to analyze, we have another problem. The problem is that every sentence, every piece of text has different number of tokens. And you have some very short sentences, very long sentences. So if you want to train a deep learning or machine learning classifier, you need to somehow to use always the same number of features to classify, to train your orange model here. You need to use always the same number of features. So somehow we need to represent any sentence with the same number of float numbers. This is what this sentence embedding component is doing in SparkNP. It will take the document, it will take the embeddings, the token embeddings, and it will do some calculations to represent the whole sentence with a fixed number of float numbers. In this case, we are using the average strategy. You see here the set polling strategy. We are using the average, so we are averaging the token representations into a single sentence representations. But you have different options in SparkNP. So here you can see, like, I feel a bit drowsy and have a little blurred vision after taking an insulin. We are able to represent the meaning of this sentence with a list of 768 numbers, float numbers. And you can see here the beginning of this list, long list. And the idea behind this at the end is that if we are able to represent every sentence, every piece of text with a fixed number of float numbers, then we can represent every sentence as a blue dot here in this diagram, and put that sentences into a kind of hyperspace, hyperdimensional space. In this case, we are drawing 3 dimensions, but in the real scenario, we will have like in a space with 768 dimensions, but you can get the idea. So the idea is that we can expect, or we will expect that those sentences that are related with adverse drug reactions will appear close together in this hyperspace, and those sentences that are not related with adverse drug reaction we expect them to be located in a different part of this hyperspace. So then we can train a deep learning or machine learning model to try to learn how to split these two groups. And once we have a new sentence, we just need to represent the sentence in this space, put this additional blue dots in the space, and see in which part of the space is falling. If it is in the left, it will be classified as an ADR sentence, and if not, it will be classified as non-ADR sentence. This is basically the idea. And this is what this classifier pre-trained model is doing. We took some pre-labeled sentences that we know in advance those what were ADR related and those what were not related with ADR, and then we train a model, deep learning model, to classify those sentences represented by sentence embeddings. And so once we have this model trained, we can feed with new text, and theoretically, or hopefully, it will classify any new text in ADR related or non-ADR related. And this is what happens when we feed with the first sentence that is related with an ADR, that this component will return this metadata with the probability of the sentence belonging to the ADR or to be related with the ADR, and the probability is very high, 99.69%. So he will classify this as a true, meaning true that it is related with an ADR. For the second sentence that is not related, you can see here the classifier is basically telling us that the probability of being true, of being related with an ADR, is very, very low, extremely low. And the probability of being non-related is very, very high. It's near to 100%. So it will classify this as a false, as non-related to ADR. Something to mention here is if you are interested just in feeding the text and getting the final classification, and you are not interested in the intermediate steps, the embeddings and tokens and so, you can, in the Spark NLP, you can package all the whole pipeline into what we call a light pipeline. It's what we are doing here in the first command. We are taking the ADR model, classifier model, and we are generating an ADR light pipeline here that then is ready to receive a text and will just return a dict Python, Python's dictionary with the classification basically. So if we feed with the first sentence that is related with an ADR, and we then feed this light pipeline with the text and using the function annotate, we will get in return, we will get the true value. And if we feed with a sentence that is not related with the ADR, we will take the value false. This is very convenient to build models in production, so you can get this light pipeline and put into a web service or whatever REST API that you want, and you will pass the text, and you will get the classification or whatever the output of the pipeline is. We can, of course, feed this light pipeline that we have built for classify with not one sentence, but we can feed with a paragraph, for instance. In this case, we are using this example that starts with I feel a bit drowsy and have a little bad vision after taking a pill, and blah blah blah. And the model is telling us that this is a paragraph that is talking about an adverse drug reaction. But we can also pass this paragraph sentence by sentence with this for loop. You see here, we are basically taking each sentence and passing to the model. And you see here, like the first sentence is classified as a adverse drug reaction sentence, but the second one that is not talking about any adverse event is classified as false, and the third one is classified as false, and the final one that is probably could be like related with an adverse event, maybe that is so far it's been very good, pain's almost gone, but I feel a bit wired, didn't have that when on 50. So this one is not clearly for the model related, it's classified as false, but we know that probably is somehow it's more an adverse event than the second and the third one probably. And this is to illustrate how can we use this output, put this output into pandas. That is a library very used in data science. And here, basically, I'm showing you how to get that output into a pandas data frame, and just print the output with the class results, the final classification, but also with the probability in this final column. And you see here that the first one is, of course, classified as a true, as an adverse event. But you see here that the second and the third had a very, very low probability of being an adverse drug event, but the final one is slightly higher than the other ones. So it means that you can use the probabilities to build different thresholds that will help you in your use cases. What if we pass this original letter from thalidomide to our pipeline? But you can see here that we are passing this original letter, and we are getting that effect, that the model is saying that this letter is related with an adverse drug event. So you can imagine you can use this for classifying your documents in your company, for classifying tweets, for classifying papers, or whatever information you want. But usually, when you are talking about pharmacovigilance, you will make questions like these ones. What are the most frequently adverse events related with the use of hydroxychloroquine? Or what are the drugs more commonly associated with gastrointestinal adverse events? You will make this kind of queries. So it is not enough just to classify, okay, you have a lot of documents and I want to identify those that are talking about adverse drug events. But you will want to get this kind of database of this column where you have the drug and you have the event related with that drug. And this is done by what we will talk next, that is the entity, the named entity resolution models that will identify which tokens are talking about drugs, which tokens are talking about adverse events, and so on. This will be another pipeline in Spark NLP that we will build to extract that information. And the structure is very similar. You can see here we start with a document assembler, but we have a tokenizer, we have BERT embeddings, pre-trained BERT embeddings, but you see here new components. This sentence detector is basically a very simple annotator that will take a document and will split the document. If it is a paragraph with several sentences, we'll split the sentence. And we want to split the sentence because for NER, we want to check the drugs and the events that are in the same sentence. So we want to add this component here in this pipeline. And then we have the core of this NER model, that is the NER, the pre-trained NER adverse drug event BioBERT model. It's trained in English. This is part of the licensed version of Spark NLP, and this model has been trained to identify drugs and identify adverse drug events inside texts. Then following that, we have this in brown, we have this NER converter that basically will combine when you have an entity that is composed by several tokens, different words, it will just put all those words together in what we call a NER chunk. That is basically for printing this information. So for the next examples, basically we will apply always the same sequence of tasks. So I will put in everything in a single function, Python function. We will fit this function with our model and with the text we want to use over the model. Basically, the first step is we will create this live pipeline from the model. Then we will pass the text to the live pipeline, annotate this text, and we will get the results. And then here you can see a couple of functions, a couple of commands that will basically extract from that output the pairs of token and NER labels to facilitate visualization, basically. So we will basically at the end we will return those pairs of token NER label that are relevant. We are dropping those that are labeled as O, that O stands for others. So no NER detected on that token. So if we pass this function, this NER model, to our thalidomide text, we see how the model is able to identify in blue the drugs and in green the adverse events, the potential adverse events. And you see this is returned in the form of a table where you have the first token congenital is labeled as adverse drug event with the prefix B. That is, this prefix B stands for begin, and then as this entity is composed by two tokens, congenital abnormalities, the second token, abnormalities, will be labeled as an ADE with the prefix E that stands for intermediate. So you have the first token chunk. The first chunk is congenital abnormalities, is adverse event. Multiple severe abnormalities is another event. Thalidomide is a drug. Distabile is a drug. Abnormalities is an event, and so on. We can apply the same for the other examples we have seen, and you can see how it is able to identify pretty well where we are talking about the drug. Even when we talk about the generic appeal, it is able to say this is a drug, an insulin, and a pill, an aspirin. And you see, and it's also able to identify most of the adverse events. But you see here that any model is not perfect, 100% perfect. So you can see here, like as GP, general practitioner, is labeled wrongly as a drug, and probably is labeled wrongly because in the context of this sentence, cool looks like a drug. Like GPs started me on 75 twice a day. So the model is confused about this structure and is considering GPS as a drug, but it's wrong. I must say that we have evaluated our models, and they are over state of the art compared with other models that have been used in open tasks. So we are about in this NER model, we are about 86 F1 score. If you want more information about these metrics, you can just contact me, and I will send you the detailed information. But the idea is that if you decide to use this library, you will always want to evaluate the model with your own data. You need to label some data and then check if the model is working for your data. So we have talked about these classifier and NER models that are good for this part of the adverse events, those that are noticed and reported, and those that are noticed and maybe are registered in the clinical record, or a patient is talking about it in a tweet, but it's not reported to the regulator. It's not officially reported. So with these technologies, we are able to identify somehow these adverse drug reactions that are noticed. But how about these unnoticed adverse reactions? How can we identify those events that even the patient or the doctor is not aware of? Well, basically, that is done by this typical pharmacovigilance or pharmacoepidemiological studies over, for example, you can take patient electronic healthcare records, and you will extract from structured data, but also from clinical notes. You will extract different drugs, the drugs that the patient is taking, and then you will extract the different health issues or symptoms or diseases that the patient is suffering. So then you can design a typical cohort study or case control study where you will compare what is the probability of having this health problem in patients that are not taking any medicine against those that are taking the paroxetine, against those that are taking the pravastatin, and against those that are taking both drugs. And as we have seen in the previous example, you will expect that those taking the both drugs will have an increased risk. Even if this is not noticed by the patient, by using this kind of study designs, observational study designs, you are able to sometimes to identify those. But how to extract that information? Well, in the SparkNP, we don't have only those ADR models. We have several different models that can help you when designing this kind of studies. We have, for example, here a posology NER model that basically is the same one than the adverse drug event model, but we are, instead of using that pre-training model, we are using a different pre-training model, this NER posology large BioBurn model. So if we use this model over this sentence here, days after the patient increases the dosage of losartan up to 40 milligrams per day, he started to develop a trunk rash. If you use on the left, if you use our ADR model, it will identify losartan as a drug. You can see here at the top, and trunk rash as an adverse drug event. But what if you want to analyze the relationship between the dosage and the severity of the effect? You need to extract also the information about the posology of how much, what is the strength of the drug that this particular patient is using. For that, you can use this posology model, where and the model will identify losartan as a drug, 14 milligrams as the strength of this drug, and per day as the frequency of the drug. In the SparkNP licensed version, you have several different pre-trained NER models and classifier models that you can use, you can combine for your specific use cases. We have models to detect signs and symptoms, diagnosis and procedures, drugs and prescriptions, risk factors for cardiovascular diseases, lab results, etc., etc. It has many different models. You can check in this link below. With demos, even you can even try with different texts and see the output. So, in summary, what we have talked about is the new pharmacovigilance methods imply the analysis of electronic health records on social media and required NLP technologies. Spark NLP is a library based on Spark that has an open source version and a licensed version that includes ready-to-go pre-training models specifically targeted to the identification of ADRs and other clinical events. We have talked about domain-specific word and sentence embeddings and how those embeddings represent the semantic using a vector approach. We have talked about this deep learning based text classifier that are able to classify if a document or a sentence is related with an adverse drug reaction or not. We have talked about name entity recognition models that are able to extract the adverse events and the drugs, and additional biomedical NER models that are able to extract things like drug posologies, diseases, and risk factors. So this is the end of my presentation. You can find me in LinkedIn, and you can find more information in these links. If you have any question.
Awesome. Thank you so much, Julio. We'll go ahead and take questions. So just a reminder to everyone, if you have any questions, feel free to submit those in the GoToWebinar control panel. And before we dive into a couple of questions that we received, I just wanted to let everyone know that in the next couple of weeks, we actually have 2 workshops coming up. So one is Spark NLP for data scientists, and that is a 2-day certification training. It's going to be half days for each one. And the following week, we have one that is specifically focused on healthcare as well. So if you haven't already, feel free to check that out. The link is in the slide. I'm also going to paste in the chat so you can click directly there. And we also have 2 free NLP summits coming up. The first one is in April, and that one is focused specifically on healthcare. And the other one is in October, and that one also has some healthcare, but also more general. So if you haven't registered for that yet, be sure to do so. It's completely free, and I pasted the link as well in the chat. And Julio, we have a couple questions for you that came through. Yeah. Some of you are asking about recording and slides. As I mentioned in the beginning, we'll definitely send that out. This session's being recorded, so you'll receive that in the next couple of days, and we can definitely include a copy of the deck as well. Another question for you, Julio, do you have benchmarkings of the pre-trained models?
Yeah, We pre-train these models. Usually, some of the models are pre-trained with internal proprietary labeled data. And then we have other pre-trained models that are trained basically with open labeled data that comes from these typical tasks, open tasks that are published year by year in the medical domain. And we have benchmarkings for those cases where there's an open task, so you can even check the benchmark. Usually, most of our models are in the state-of-the-art level because we are using the latest deep learning architectures. If you want more information about that, just email me and I will send you the detailed information about the benchmarkings. But our recommendation always is don't trust on the open benchmarkings, because you are never sure that if someone is tricking the benchmark. So what you always will need is, okay, I have my own corpus, labeled corpus gold standard, and I will check against that corpus any model that you want to do. Because sometimes as you know, if you are use NLP or the machine learning, models work very well for some data, but when you use in other data, it will work equally well, or it will work worse, and then you will need to train your specific models, something that can be done with the Spark NLP. You can train your own models with your own data. So basically my recommendation is, yes, I can send you the benchmark, but probably you will want to check against your own gold standards.
Awesome. Thank you so much for that answer. And if there's any other questions, feel free, as Julio mentioned, to reach out directly. And thank you so much, everyone, for joining us today. We hope to see you on the next one and in our upcoming events as well. Thank you all.
Thank you. See you.